Assalamu alaikum. Hi guys. We're going to discuss molecular biochemistry. Basically, we're going to start with the basics of DNA. The first thing that comes to mind when it comes to molecular biochemistry is DNA. So, basics of DNA. Now, what is DNA? It is basically the genetic structure, as we know, genetic code. It's a basically basis of human life or human cells contained in the nucleus of eukaryotic, or we can say human cells. And to further remember what it is made of is nucleotides. So we say it's a polymer of nucleotides, which is the basic structure of it, right? Now, what is a nucleotide? Or first we come to the image of it. We have this looping structure, right? Two strands looping around each other with the bases, right? The basic structure that comes to mind is this. There's like a ladder with its rungs, right? So this is like a DNA double helix that we name it as double helix, right? So what is nucleotide? This nucleotide is basically consisting of a sugar backbone with the nitrogenous base attached. It's a bond and a phosphate group. And it needs to be differentiated from nucleoside at the same point with the S nucleoside, which again has the ribose sugar with the base but no phosphate. So there is no phosphate group in nucleoside, which is basically the precursor of nucleotide. So when the phosphate group attaches, it forms the nucleotide, right? Further, for the nucleotide, we're going to discuss its bases. We have 5 bases: adenine, thymine, guanine, tyrosine, and we have uracil, right? Now, 2 of them are sorry, I haven't written with the same, but I will show this way. 2 of them, these guanine and adenine, these 2 are known as purines, and these 3 are known as pyrimidines. Pyrimidine has 1 ring structure, and purines, the shorter name, has 2 rings. Just the basic thing to remember. Now, this is the only base part. When it is together with the phosphate group and the sugar backbone, the sugar, it will be the sine part will be there. There will be adenosine and something like thymidine, guanosine, cytidine, uracil will remain uracil. So this is a little basis to remember of DNA, how we call it as a polymer of nucleotides, right? Nucleotide is basically the ribose sugar. In case of DNA, there will be no H group. This H group will be missing. There will be no H group. There will be only one OH group. So that will be referred to as deoxyribose. It won't be a ribose sugar anymore. It will be deoxyribose sugar. So there's a little thing to remember about the sugar backbone here as well. It won't be a ribose sugar that is mainly present in the nucleotide, but it will be a deoxyribose sugar with a nitrogenous base that we have discussed. That there are 5 bases, basic 5 bases, with the category purines and pyrimidines, and with a phosphate group attached. Now it depends on how many phosphate groups to attach. If there are 3 phosphate groups attached, then adenine will become ATP. It's also a thing to remember. So this was something about the nucleotide. And now I will go through the chromatin structure basically. So when we say DNA, we say it is composed of chromatin, right? It is composed and condensed form of chromatin that fits into the nucleus. So what is this chromatin now? Chromatin is just DNA plus histone, right? With a complex structure. Histones are the proteins. Some things to remember of histones is that it's a protein. They are proteins with 8 subunits, and with an external subunit that is H1, that basically links one nucleosome to another. And this histone protein has basically it's basically having 8 subunits with the different names like H2A, H2B, H3, and all these subunits with H1, that's the external part, that basically at one point it attaches to the linker DNA, and it has stabilized the chromatin structure. This is very important in a histone, that stabilizes the chromatin fiber, not let it just lose in the nucleus. So this is the basic structure of chromatin. When it comes to 2 types of chromatin, we say euchromatin and heterochromatin. What is euchromatin? Euchromatin is more of a loose chromatin that is undergoing histone acetylation, and it is more prone to, or it has high chance of transcription. So this is the part of DNA we say, or this is a part of chromatin that undergoes transcription or division, or you can say all that, basically whatever is, it's bringing one DNA to another form, right? So this transcription is crucial when it comes to euchromatin, because there is high rate of transcription, as there is high rate of histone acetylation going on in this area. Why? Because it's more of a loose and relaxed chromatin. Means it allows the DNA to transcribe easily. How it looks is that there is this octamer histone with 2 loops of DNA. Okay, so there are 2 loops of DNA. There is 2 loops of DNA with this is a linker. This is basically H1. This is the subunit histone, 8 subunit octamer, and there's 2 loops of DNA, and this 2 loops of DNA is attached via another. Now this whole thing is known as nucleosome. Now this whole nucleosome attaches to another nucleosome, the same structure, and this whole is euchromatin, more of a loose structure. When it condenses more further, it becomes heterochromatin. Heterochromatin is basically more of a condensed and preserved form of DNA, right? This is the part that appears darker on electron microscope as well. When we view nucleus under the electron microscope, the darker parts of it will be the heterochromatin, because it is so condensed and preserved, right? There is increased methylation, DNA methylation going on here, methyl groups attaching, that preserves the DNA, that basically makes the transcription inactive in DNA, and there is less acetylation, histone acetylation going on. So this is very crucial DNA for a human body, because it preserves like 70 to 75% of the DNA structure overall that has been in the nucleus. Like 70 to 75% will be preserved, and it will be not undergoing any transcription, it will be more of a controlled form of DNA that is preserving the DNA itself. So this is mainly about the structure of DNA. DNA is basically composed of chromatin. Chromatin is DNA plus histones. DNA is a helical structure. It's a double helical structure with the basic structure that it has is nucleotide. Nucleotide we discussed has a sugar base, has a sugar backbone with a nitrogenous base, and nitrogenous bases are 5: adenine, thymine, adenine, thymine, guanine, cytosine, uracil. Uracil basically comes, sorry, it just got off. Uracil basically comes in RNA, not DNA structure. This is one of the bases that is present in RNA mainly, not DNA. And adenine over here in the helical structure will bind with guanine with 2 bonds, and thymine will bind with cytosine with 3 bonds. It's more of a guanine will bind with cytosine with 3 bonds, and adenine with thymine 2 bonds. This also has importance, right? Everything is just wiped off. Sorry. So guanine with cytosine and thymine with adenine. Just remember that A, T, and C and G. CPG islands we call it because it's more of a stronger bond and it's highly present in the DNA methylation because it's more of a stronger and more of a preserved bonds. There are 3 bonds, so it's stronger definitely. So this also has significance. We'll further discuss with the pathology of like SLE and psoriasis. That is important when it comes to DNA methylation, when it comes to preservation of DNA and its structure, why is it important. So yeah, it's interesting. So this is the first part. We'll end with the basics of DNA here.